Thanks for tuning in. This week's TechTake always starts. The National Computational Infrastructure Technologies celebrates and pays our respect to the Ngunnawal and the Ngambri people of the Canberra region and to all the First Nations Australians on whose traditional lands we meet and work and whose cultures are among the oldest continuing cultures in human history. I'm Fred, training officer here at NCI. Today, we are very pleased to welcome Sonika Tyagi from Monash. So Dr. Sonika is the adjunct lecturer at Monash University and the machine learning leads Superbug AI at Alfred Health in the Monash University. So today, Dr. Sonika will give us a talk about the challenges and applications in the digital health area and the computational biology. Without further ado, let's welcome Sonika. The floor is yours.
Thank you, Fred. Thanks for your kind introduction. Thanks for having me here. I'm going to talk about some applications of machine learning that we've been employing in the area of digital health and computational biology, and we'll talk about some of the challenges we have come across and the solutions that we are implementing. So I mainly intend to talk about various biomedical data types that we deal with, common applications in our research work of machine learning applications, and talking about multimodal data integration towards the end. So as we know, like all other fields, deep learning has a huge potential in the field of biomedicine as well. And we are seeing the biggest impact in the area of precision medicine, which specifically focuses on understanding and treating diseases by combining this various type of information that we have available for individuals to make patient-tailored decisions. And this is a data-driven approach. And the common type of data that we use here are molecular data, which is also known as omics data, which is generated from various biomolecules in a high throughput manner. The other type of data are clinical data and health data collected through electronic medical records in the hospitals, from wearable devices, and so on. Now imagine that an individual with a complex disease condition turns up at a clinic, and a domain expert might order some genome sequencing to match with the database to find any disease-causing mutations or disease-causing genes in their genome. They might also perform some pathologies or do some other sensor measurements. They might have access to other lifestyle and environment data for that individual. Now you could use a machine learning algorithm to combine all of this data to find all those non-linear relationship and correlation between different factors to be able to do this individual specific prognosis, diagnosis, and treatment plan. So this kind of approach not only will have a tailored care plan for the individual, but you could also predict future risk of developing any conditions, or the risk of readmission to the hospital stay, risk of ICU admissions, and so on. So this helps with not only a better care plan for the individual, but also helps healthcare facilities to prioritize their resources if they have access to these risk forecasting for the people that they're caring for. Now, looking at the data specifically, this comes in various shapes and sizes. So the majorly, I can divide this data into structured and unstructured data. Structured data being the tabular data, which is usually the quantitative numerical measurements that are recorded for different observations and different variables that it's computed for. The other other category is the unstructured data that involves various subtypes such as images and documents, various codes that are used for diagnosis or procedures or even instruments. There may be, clinical notes available that are written by nurses and doctors. And then there is genomic sequence, which is just the sequence of DNA written as by using 4 letters, ADGC. So these are just long string of text that we are looking at. And this data is being generated in high volume at very high velocity, and it's obviously of multiple modalities and multiple dimensions. So you can imagine that each of these data type will require heavy pre-processing to be able to use for their downstream applications. Now, let's look at the molecular data, which is mainly being contributed from the field of genomics, and it involves majorly the DNA sequence data. And this has been growing at a very fast pace, leaving behind even the data coming from astronomy or the social media platforms. And this is mainly because of the technology improvement in the high throughput sequencing arena, as well as the dropping cost of sequencing, and that has been the main reason of the growth in this space. Now, taking you back to the central dogma of life that states that information coded in DNA can be transcribed into RNA, and then RNA can make proteins that are the functional building blocks of life. So if I represent a genome, which is the entire DNA content of a living being, as this green circle, then majority of this information is transcribed into RNA and what is called as transcriptome. And now then part of this transcriptome can code for proteins, and hence it's known as coding RNA. Or this is also known as the coding part of the genome, and the remaining transcribed part is known as the non-coding part of the genome, which is all RNA. And some of these RNA have structural role, and many of the majority of the RNA have other functional roles in gene regulation. And this is like a more recent focus on these molecules for having their critical role in various disease and development processes, because they will regulate how genes function. So just imagine that all of the cells in a human body has the same set of DNA. Then how some cells, look and perform differently to the other cells. For example, all the cells in heart will have, similar sort of function. They'll have a set of genes that they express and use, as compared to, let's say, skin cells. So this is so the analogy that I can give here is like of a recipe book, that you have a book and it has all those different kind of recipes, or some are only being used for breakfast, some for lunch, and some for dinner. So how these different cell types use this selective information on genes is regulated by a sophisticated molecular regulatory mechanism. And this is where these this is like relatively newly discovered roles of these RNA, that they are part of that regulatory machinery, and hence are very, very important for understanding their function in different biological processes. Now let's look at how different layers of information is generated by these high throughput sequencing technologies. So human DNA is very long. It's made of 3 billion base pairs, and this needs to be stored in a cell nucleus, which is on like nanometer scale. So the DNA has to be really packed into a compact form called chromosome to sit inside the nucleus. But in order to read the information on that DNA, the DNA needs to unpack and expose the genes that can then make their product in the form of RNA, and then RNA, making protein, and then these proteins carry out various functions in the metabolic pathways. Now, how this DNA unpacks is through various signals that can be captured through these are just the names of different sequencing technologies in these gray boxes. And as you can see, they can capture different type of information from this layer of what we call as regulatory signature. And then all these functional molecules, their RNA, protein, and metabolites, they make up a functional signature of in a cell. So using these various high throughput technologies, we can generate this data at different level. So you could have data, coming from DNA, RNA, protein, or metabolites. So you can imagine the different types of data and the dimension and the size that we are talking about. So you could generate this millions of data points from each experiments, and this can be done for different tissue samples and for different individuals. So the data volume grows very, very quickly. Now this data, just by the sheer size and the complexity of it, I know has gained a lot of attention from application of various machine learning algorithms, and the applications that these algorithms have in the genomic space, I can categorize them into four main types. So first one is pattern recognition. So you're looking for a particular pattern, such as the gene or some recurring signatures like motifs. You could be looking at solving these 2D and 3D structures of these biomolecules, such as DNA, RNA, and protein. You could be doing a supervised learning task to train a model and then predict or classify unseen data. Would also apply these algorithms for doing image analysis. And the neural network architectures that are used here are mainly, are either fully connected neural networks that assume features in no particular order and have been used for pre-computed features generated from this genomic data. The other type of neural network architecture that has been commonly used is the convolutional neural network that assumes that features have some local, pattern, for example, the subsequent DNA basis. And then this convolutional network has been used to find local patterns in DNA sequences, protein sequences, and even in images. On the other hand, recurrent neural networks, they assume that the features are to be processed in a sequence, and the state of the current feature has some memory of previous features and has been suitable to analyze time series measurements as well as DNA and protein sequence data. The other type of architecture is the graph convolutional architecture that assumes that the features that you're using here have some prior relationship defined by a graph, and have been these architectures have been used in studying protein-protein interaction networks, citation networks, and even protein structures. And more recently, they're gaining a lot of attention to predicting missing data, which is a very common problem in the healthcare data, as well as predicting new patterns that were not part of the training data set for machine learning models. And I'm obviously missing some of the latest implementations of that is of like transformers and autoencoders here. And I'm pretty sure you all must have heard of this new, revolutionary application of deep learning in the biology space. That was the AlphaFold by Google's DeepMind, which was a clear winner in the 14th critical assessment of structure prediction competition in 2018 and 2020. So this algorithm predicts protein structures, which has been a long-standing problem for last 50 years. So the standards are to predict accurate structures are by using X-ray crystallography and cryo-EM, which are obviously very expensive and very, very tedious experiments to perform. So here, AlphaFold can help in that space to predict new and accurate structures. It has already been applied to human proteins. There are like 20,000 human proteins, and more than 98% of the structures have been generated using this database as well, this tool, as well as they are making it available as open database, and the source code has been available as well. Next, I'll talk about some of the applications that we have been implementing using machine learning algorithm. So first one is in the pattern recognition space, which is to look at the gene regulatory motifs. So as I said earlier, for a gene to turn on or off, it relies on the regulatory machinery that works around it. So one of the components of this machinery are these protein regulatory proteins that will bind to the gene's upstream region in a sequence specific manner. So they will recognize these different patterns where they can bind. So they don't always bind to an identical pattern. So they can bind to very, very similar, like closely looking, these binding patterns on the DNA sequence, which can then be represented as a consensus motif. So that is one feature that we need to take into account. The other thing is that these motifs usually work in combination with two or more neighboring motifs. So there has to be some optimum distance between them. They might be able to interact with each other either directly or through some cofactors. And the challenge here is that these binding sites are very small. They're tiny as compared to the entire genome, which is like billions of base pair long. These patterns are only like 5 to 12 letters long. So finding them by chance is very highly probable when you're running an algorithm. And the other thing is to find this all possible combination and how many are working in combination. So finding those clusters of motif is another challenge. And so the first problem of just detecting individual motif has been a very popular, genomics problem. And there've been lots of wonderful solutions that exist there, and we also have implemented one algorithm there. But the problem that today I'm discussing is to find the cluster of these motifs. So two of my master's students, they implemented this algorithm to using a convolutional neural network architecture, where we are trying to find these two consecutive motifs, and we take into account some neighboring sequences as well, because the structure of this DNA in like 3D, you can imagine this DNA will have a 3D structure, and the structure will determine whether protein can bind to it or not. So all of this information is concatenated through a process called feature concatenation, and then is one hot encoded. Then this data is one hot encoded, and then different filters are applied on these different convolution layer to find a match with the filter with the position, and then the typical CNN architecture. Then we have activation function, which is relu in our case, and then maxpool will then collect all these average of different filters to have input going through to next layers. And then we have applied hyperparameter tuning using Bayesian search to find this optimum set of parameters for the model. And then we've been able to successfully develop this model and applied it to a database of containing more than 321 motifs generated through more than 2,000 samples. We have predicted clusters and have done annotation, and have made this available through our web server. The other application that I'm going to discuss is for predicting these non-coding RNA structures and functional motifs on them. So as I said earlier, those small non-coding RNA are one of the molecules of significance in the gene regulatory machinery. So these are around 100 or so base pair long, and they can fold into a stem loop secondary structure. And then finally, from one of the arms, the actual functional molecule is spliced out, and this guy can go and turn off a gene. It'll go and attach to a gene and will turn it off. So it's very important to know these these molecules and what genes that they can target, so that you can manipulate that in like genetic engineering to target these molecules and see how it affects its the genes that it's blocking. So our aim here was to predict these structures, because these structures are very, very important for their function. So here what we have done is we applied stochastic context-free grammar, which is one of the natural language processing technique. So we developed grammar to understand this folding, and then developed a probabilistic model that learned these probabilities from known data, and we were able to predict unknown new structures of microRNA, and we have successfully applied this to predict new microRNA in skin cancer or melanoma. I also had a summer project where with very exciting batch of high school student that came and worked with me and extended some new utility in this pipeline to be able to predict these splicing sites for splicing out this mature microRNA from the stem loop structure using a feed-forward loop. And the project was very, very successful, and they came and presented the work with me at the Launch Genome Conference. The other type of long non-coding RNA that we're looking at are the long ones, which are longer than 200 nucleotides. So these are some structural and functional properties that we are using in building the deep learning models for predicting these RNA. One is their location. So the green ones are the coding genes, and the pink one are the known coding genes that we are interested in picking up. So they may appear in different location. So they may be in between two coding genes, or just on opposite strand of DNA in the opposite direction, and in an overlapping situation or non-overlapping situation, or they may be embedded within a coding gene. So we are taking all of these features into consideration. The other one is their functional features. So these long non-coding RNA that are shown in blue in this cartoon diagram can interact with other protein and DNA. And this structure of these RNA is very, very important, and these contact sites are the motifs that are very important to determine where they will go and interact and how they will function. So we are capturing all of these information into various feature sets to build our model. So we categorize these features into three sets, so which is the sequence-based feature, structure-based feature, and those interaction features. And we were also interested in learning what are the features that are common across different species, and if we can develop a generic model or a species specific model using these features. And we were able to do that. We use these features in a fully connected artificial neural network model, and we're able to develop different versions. So one was human specific and species agnostic model, and we were able to apply species agnostic model to different species as well. And it turns out that these RNA have lots of common feature across different species groups, and that could be utilized into this model. And we have this again, this model available through both as standalone tool as well as a web server. Then we were also looking at just a sequence-based approach where we don't have to compute features, because all those features may not be available for all of the species RNA that we are looking at. So we looked at another an existing model actually, which was based on RNN approach. So it just buckets sequences into different length groups, predict a particular feature called ORFs. We'll do a one-hot encoding, and then put that through this LSTM architecture. And then we realized we wanted to use this as an option in our pipeline, but realized that we had to do a lot of re-engineering and recoding before we could do that. And now we made a lot of even improvement over this algorithm, and this can be an option in our pipeline. Just to summarize some of the challenges that we have come across thus far was one of the, probably a common challenge that we see in supervised learning is the lack of label data. And also there, we rely on fetching data from these public open access repositories, and sometimes there are discrepancies because of lack of standards in naming the sequences between different data sets. There could also be data privacy issues if we are trying to access patient data, which can, block some of this information for us, and that leads to, just overall lack of, usable data for a particular study design. We also realize that if you're trying to reuse some of the existing models, there's lots of challenges that we would come across. One is, just lack of generalization, because we don't have access to the data and the code and all the, hyperparameter tuning validation strategies that went into developing that model. So it's very hard to know how this can generalize to our experimental condition. We also learned that, a lot of these models are available and are not being updated and maintained, and may have like outdated version of, software that was used in environment that was used. Like in this example, we had like code developed in Theano environment, and we're using Python 2, which couldn't be just reproduced straight away after downloading the model. Especially as you can see, we are normally looking for rare small pattern in a huge amount of genomic data. So there's a lot of, combination that to test, and that require, bulky models and, bulky compute to run these models on. Again, in biology, there are a lot of, variations that exist, and a lot of different combinations, of experimental condition where you want to validate your models, and you may not always have that validation ground truth data available to assess all different scenarios. So this is just an open challenge. The other one is, to be able to process this large amount of information, setting up algorithms that can run these things in parallel, and to use high performance compute or individual high performance machines. Again, there are a lot of gaps in that space, and you don't always have algorithms that will adapt to a compute environment, and vice versa. And as I said before, these models, have to be of reasonable size, and they should be portable, and should be able to scale to different tasks. So normally we are developing models for very, very specific tasks. And if you want to, solve a different problem, you will again have to start from scratch, building an idea, code for it, and then do different experiments to optimize the model. So can we, take like a more modular and layer by layer approach, where different layers can have specific purpose that can then be transferred to learn a different type of pattern. Interpretability of deep learning models is another open challenge of the field, that we are able to get better predictions, more accurate predictions, and be able to handle these non-linear relationship from large amount of multi-dimensional data using deep learning algorithms. But what we also lose is the interpretability of what are the predictors and that have most impact on the outcome of a model. And this becomes very, very important when you are applying this for clinical application, that we need to understand what are the things that are affecting a model's performance in either direction. Now I move to the next section of the talk, which is on data integration, or what I call is multimolecular data harmonization. So first I'll talk about these omics profiles and DNA sequence harmonization approaches. So as I said earlier, the using these various biomolecule high throughput profiling techniques, we can generate different types of omics data from different layers of information. So for example, you could get these signals from gene level, protein, and metabolite level. And as you can imagine, in a disease condition, a single type of data, just by analyzing single type of data, cannot give you the full information, full system view of the disease, because it's when something goes wrong, there is a cascading effect at different layers, and there's a lot of correlation and relationship between these changes. To understand what went wrong, you'll have to analyze all these different layers together to understand these markers that can be used to develop therapies and treatments. So by merging this information, you could also uncover novel information that was unable to be revealed just by using one data type. So zooming this out further, and you could be looking at this integration of information at different layers. So looking at protein interaction, drug interaction network, non-coding RNA regulatory network, disease association, and zooming it out to looking at healthcare information. So you can get an individual's a holistic view of a first person by overlaying this different type of molecular information as well as healthcare information to get a comprehensive understanding of a condition that you're studying. So when we started to look at this data integration problem, we looked at literature and what exists out there that we can reuse for these multimodal data integration tasks. So this was a review done by my PhD student, and we realized that there are a lot of the tools that are published as data integration tools may have different meaning in different contexts. For example, a lot of the data integration tools are just looking at analyzing these individual modalities separately and then combining or aggregating results at a later stage. Another category of models was to just look at batch correction sort of strategies where you have the single modality of data will just run at different times and different experimental conditions. What we were really interested in to have models that can take information from different modalities. They're generic enough to take information from different modalities and then combine that in a generic way. The other condition that we had was that we are able to use data into least processed form possible, because as you process the data, you have risk of losing some quantitative information from that data. So that's the gap that we were working in. And we have started developing some methodology. So one of the method was to integrate this multiple omics type of data, and we use this for looking at some COVID data that was available from 2 different studies on 24 and 100 samples, and then we were able to integrate that data to find out some new targets for COVID-19 therapy, and the pipeline is published as well. So in this case, we discovered some drug targets, and also another intern student, she ran some analysis to look at the suitability of these drugs, and we realized that some of these were already in clinical trial. So the just to give you an idea of what this pipeline takes as an input is just these measurements as tables. So these are quantitative measurements maybe coming from different omics experiment. It doesn't matter what omics experiment that you have, as long as we have these matched samples there. And then in the end, it generates these low-level correlation between the features. Now that we are able to have these correlation between different functional features, we wanted to overlay this regulatory code over these features to find relationship between regulation and the functional signature. So here we are utilizing natural language processing techniques to process these genome as a large piece of text and try and find these small patterns from these texts that are relevant to our condition. So if you look at a typical natural language processing workflow, so you have an input sequence of text, then this is broken up into smaller subunits. Those subunits are then converted into numerical form of vectors or matrices, and then these are modeled and used in downstream tasks. But for biological sequence, this is a challenging area, and we realized there are no packages that were available to where we can do this bit of processing to convert sequence into smaller chunks, and what those smaller chunks should look like. What is the the best strategies or more optimum strategies for transforming that data into numerical form. So we have developed, a new library of functions where we provide different options to process genomic data into all these different transformation level. And watch this space. That we will be publishing this soon. So the part I was talking about is that normally, natural language processing tools, for example, the one that are based on English language. English language already, has, words, spaced by this space character, and you can easily, break them up at word level or character level and then model it. But English language is more like, maybe Japanese or Chinese language, where we don't have any gaps that we can utilize now, but we don't know what word size to use. So for example, you may fix a word size and break it up into those word sizes and then use those as, analogous to English language words and process. So this is another area of investigation. The other strategies that we are applying are purely data driven. We let the algorithm decide what will be the best split point and then generate these words of different lengths, and then we are using it. So we have developed this entire suite of functions that are being used to process sequence data. We have applied various machine learning models as downstream application of that genomic data converted through NLP processing pipeline. And this is an example of distal bird, where a group of sequences were classified using this NLP approach. And it's just a nice screenshot of the 1DB sweeps. And I really like it because it gives you a nice view of the progress and the system usage and everything in one place. So we've been able to apply that to our data set, and we have been able to detect some important patterns from genome that were significant for this case study of sepsis that we were looking at. So we have now a combination of genomic signatures and the functional signature of the sepsis condition, where we were applying these methods. And just very quickly, looking at the healthcare data. So healthcare data that we are accessing is through hospitals' electronic medical records. So you have an individual's, records of like immunization, vitals, pathology, clinical notes, prescription, other admin details, and finance details, medication. So all of that information is digitized and accessible that we could use. So as part of the Superbug AI project, we're looking at individuals with bloodstream infection, and these pathogens that cause the bloodstream infection can sometimes be antibiotic resistance. So in this project, what we are trying to do is to do a comprehensive modeling of the host and the pathogen interaction. So we collect all of this electronic medical record data for the host, and we have this genomic data coming from pathogens and generate features from that, and to then model and ask different clinical questions. So some of the examples of I'll get into that in a bit, but I wanted to point out here that this data is very, very messy and very, very hard to analyze, because as you can imagine, you don't have same level of measurement for all patients in a cohort. They may be coming at different times, so data is very irregular and episodic, and so just dealing with a lot of nuances in the data is a lot of work. So one of my PhD student has developed this pipeline to do this quality control on the data, to find missing data and outliers, suggesting pre-processing techniques to process this data and transform this into data metrics that can be then used in machine learning models. The other thing is that these electronic medical records data are mainly available as relational databases. So we are also adopting common data models for those database structures, so that data from multiple sites can be transformed, and then same set of analytic methods can be applied on those methods. So the quality control, along with adopting standards for the data, are the way to be able to analyze this data and merge across different sites. So this is an example of a clinical question that we are asking from the data. So we use vitals and lab measurement for individual infected with these bacteria and having bloodstream infection, and we are predicting mortality risk by using an ensemble of representational learning methods. And as you can see, these models outperform the baseline model that are routinely used in clinical practices. This is another example of using the unstructured data, which is from clinical notes. So clinical notes are very, very rich in information. They have lots of records of patient history and different information that is recorded by different nurses and doctors at like pre-admission, admission level as they progress through and they're discharged from the hospital. So, but then because this data is also like handwritten, there's a lot of issues with processing this data, because they not only there'll be like grammatical errors, they won't be like sometimes proper sentence structure. There also a lot of abbreviation that are used. And so data is very unstructured and irregular that needs to be processed, and we've implemented a pipeline to process that data, and asking different clinical questions, for example, predicting unplanned readmission within 30 days of discharge. That's shown here, an ROC curve with the model and how well it's performing. So next we are moving to data integration in this project, and to overlay information from other components, as I discussed earlier. And we have already implemented various strategies for data integration, and have a good idea of what works with what type of data. So eventually we are working towards like a universal sort of harmonizer that is generic enough to accept information from different modalities, and is functional and robust. It's usable by other people, and is also interpretable. So these are the features of the harmonizer that we are working on. So just to list a few challenges that we have come across in the data integration process. So I wish we can start sharing models as we share code in the. I'm a pro open access open source tool, and I wish like we now also focus more on sharing models in a way that these are reproducible by others and reusable by others. When we are using these models, in absence of training and data and the validation strategies, we may need to adopt some empirical models to evaluate the generalizability of these models to be able to use in other applications. In data integration, multitask modeling is one of the approaches, and then that we realize that it requires lots of multiple loss optimization in the machine learning modeling approaches, and that's another area of investigation for us. When you're combining data, as it is, deep learning models are known to overfit when you are using large amount of data, but having contamination because of various batch effects can also lead to spurious outcomes. And like other data sets with this health data and biomedical data, the biases are natural. You tend to have access to data from more and more, like people who are very, very sick. The data may be biased towards particular gender or population sampling, and those are things to you need to be aware of, and these need to be accounted for. Very, very critical in at the data modeling stage. So with that, I'd just like to summarize my talk today, that new high throughput research and development activities are generating data at scale, and we can easily say that biomedical data is big data. And this also poses new fundamental challenges for machine learning algorithms. Genomics often have correlated data for common biological activities, hence integration is of different type of data can provide a system level view. Data integration will provide like a holistic view when we're looking at like infectious disease situation. Advances in machine learning is bringing exciting new advancement in this space of integrative approaches in digital health. And with that, I would like to thank people who I've worked with all these years, and people and all these funding agencies that have funded my research, and all of you for listening.
Thank you, Sonika, for this wonderful talk. I think you covered quite a wide, broad range of topic in this hour. I'll go just through a couple questions I had while I was listening to your talk. The first question is about, so you mentioned the AI training models, and from my very preliminary impression is normally you will need a rather large data set for that sort of is critical to the accuracy of your training. Is my understanding correct that you will need real human genome data? And in that sense, how concerning the upstream, how those data are accessible? Are they accessible to the public, or they are quite restricted? And are they from clinical institutes, or are they from this centralized library? Or yeah, that's my first question.
In a perfect ideal scenario, if this data was centralized and was accessible for research purpose, that would be perfect. But obviously, that is not the case because of the privacy issues. So data, all the human data is accessible at different level by different means. So for example, in our case, we had to get a special ethics approval to get access to the cohort that we are studying. And similarly, data is being shared across institutions as well. So this is very, institutional level policy, how the data is shared from that space, where data is very sensitive. But then there are also big consortium like UK Biobank and TCGA that are making data available. So again, I know there is a procedure that you follow and can get access to this huge amount of very, very valuable data. So I would say in a broader sense, this data is accessible and open to be used for research purposes.
Right. But yeah, like I say, you probably have to go through those efforts to apply for ethical approval, these sorts of things. Oh, right.
Yeah. The other thing is, yeah, the second part of this is, where do you analyze this data? So normally, we bring the data to secure working environment and analyze this. But maybe in future, we take models to the data and then run it there. So those are, that's another side of the story from the practice point of view.
Right. So how large? Yeah, again, I'm not from this place. So what's the sort of matrix you have to measure the size of data? Like how many people's genome, or like, yeah.
Yeah.
And how do you measure that?
So for example, in the current cohort we looked at, we had like 5,000 patients data that we were looking at. But if you look at the bugs, for those 5,000 patients, they may be like 3 bugs per person that, that they, contracted, and then we have genomic information for. And the genome is sequenced at like 10x level, which is like 10 copies of
Right.
that needs to be like. So this, this just start extrapolating, very, very quickly to exponential scale. It is, good,
Large data set.
sizable data. Yeah, yeah.
Right. Okay. Yeah. Interesting. So this is one question from Philip. So may I just repeat the question? Philip was asking, you covered a lot of projects. Just curious, how large is your team?
Yeah. So obviously this was not just current work. So this is over past 3 or 4 years, and some of the contributions were from master students and PhD students. So they work with me for a period of time. So there's also a lot of turnover in the team, but currently, I've got like 4 PhD students and a couple of master students that are working with me.
Okay. Meanwhile, I have a second question to ask you. So you mentioned about pathways again, because it's such a broad um, um, topic. You have not just the AI part. You also covered this clinical usage. Especially, I think you used the words um, uh, harmonizer. Is it right? Harmonizer is this the integration platform. So from what I understand, the in our at least in Australia, the digital health um, transfer, transformation is quite us having some issues even with some current platform. For example, I'm thinking about the My Health Record. Even this has been pushed and it's part of the MyGov, it's a national wild platform. Even with that, a lot of doctors for various reasons are not, the typically tech savvy people, and they're quite reluctant to use those digital platforms. Um, so would you have any comments on um, how difficult is to apply those applications, clinical applications, in this of the downstream in the clinical usage?
See, what you're pointing at is a very critical gap that we currently have. So when we develop model, we want to validate the model's performance in different conditions. So ideally, we should be able to look at not only the one that we have access to and we are generating our models for, but also to look at some unseen data that can be used as an like independent test of a model. But that usually is not, it's not easily available. That kind of data is not easily available. So but things are improving. We have like a digital health roadmap for different states and in Australia, and we are trying to make access to data for research purposes easier.
Yeah,
But
yeah.
at the moment, this is a gap that we have, and there's a lot of hurdles that you have to jump before you can get access to data and start using it.
It's a broad range and it involves the all sorts of parties and for this integration.
I can take the next question. Yes, so Philip, your question is about applying these standard ontologies. So definitely, this is part of pipeline. So yeah, sorry, I missed that bit when I was talking about the data standards. So yes, we do annotate data with standard vocabulary, and this helps us a lot to remove a lot of anomalies in the data. So sometimes units may not be homogeneous or incorrect, and just using standard names for medical concepts, it become it's very, very important for to harmonize data together. So yes, we do use nomad and 84 other vocabulary that are part of a system. So we get it from Athena vocabulary platform, and apply those to our data to annotate data and before we start using them in our models.
In page 15 of your presentation, you show neural network. And in that one, you are encoding the nucleotides using a sort of matrix. So you will have like your A, T, G, C, and each one will correspond to one quantity in the matrix. Do you take it just as ones and zero, or is like the probability of having a C versus the rest? Or how do you actually implement that?
Yeah, so we used standard one hot encoding. So this is basically zeros and ones here. So we have only 4 letters in DNA, A, T, G, C. So where at position where we have A, then you have 1 there, rest of them is 0.
Yeah, because in that case, I feel some of the patterns you will observe there will be, in a way, you can reduce that because every time you have an A, all the others will be zeros. And with the rest will happen the same, which is not what happened in an image. You normally have all the pixels have a certain value, right? So instead, I believe will be possible to encode it in only 2 numbers. So you have one to define, for example, A's and C, and the second column to define whether it's the reverse complement. So you will have like T or D, depending how that encodes. And that might also help with the training. So you don't have that many empty pixels, let's say.
Yeah. So in the current pipeline that I was talking about to process genomic data by different tokenization methods and numerical conversion. So we do have all those options. So we have like this ordinal approach where you can give different numbers to different letters, and then we have one hot encoding, and then even word embeddings and more other like transformer based approaches that are available as options now.
Yeah. The other question is about the part where you are using the natural language processing. And in that case, you mentioned the difficulty of finding the right size for the words. And you mentioned you can use, for example, a fixed length. But how likely is that is to be? I mean, you might, like in a natural language, you have words of different size. And I don't know if there is a way to give that flexibility, because you might have motifs that are only 2 characters, or you might have some that are 100 characters. We don't know, right? So I think that's
absolutely
quite challenging.
Yeah, no, completely agree with you, and that's an issue. So in practice, what we have used is a range of word size. So like let's say from 3 to 8, but still may not cover the entire scenario. But the other thing is, the feature space starts exploding very quickly because you have 4 letters, and if you take like Kmer of length 3, then it's 4 to the power 3, a different combination, and then you try that for all different length combinations. So the feature space really becomes very big, and then we run into this issue of reducing the space. So that's why this is the approach that we realized from our experiment that for smaller data set, this can still work. But when you are dealing with larger scale modeling, then we need to look at other options that also we have implemented.
Okay, thank you very much.
No problem. Yes, Aslam.
Hello, I have a question related to the slide 34 about NLP pipeline. I think that you mentioned that you don't find enough tools in order to convert the input sequence to subunits, numerical subunits. I'm just wondering, have you tried to use sliding window technique in order to, I'm not sure it's going to be merged in your pipeline or not. The sliding window technique can give you kind of calculate some numerical based on the sequence. Yeah. Is it?
So that Kmer and Gram approach is exactly that. So basically, you look at a window of size and extract all the k-mers of length that you're interested in, and then build the final n-gram lookup table. So
Okay.
that approach is basically, sliding window based.
Okay. There's one more thing. I'm not sure it can be also useful for this work or not. But as I understand, there is a kind of periodicity in dinucleotide for the DNA, especially in some specific part related to nucleosome and others. So have you tried, or is it possible to embed it in your tools? I'm not sure about it. I'm just trying to ask you about it.
Yeah, so definitely the context is very, very important, and defining that context is also important. So when we were looking at these small, regulatory motifs, then we are looking at, some flanking regions around it to take that into consideration, because the sequence will determine the 3D structure of DNA, and it's important to understand, are there any structural features that will determine whether a protein can bind here or not. Similarly, when you're looking at the, you're modeling the entire genome using NLP. So positional embedding is one of the solutions that we are able to capture that information, especially in the distal BERT architecture. By default, it has the attention mechanism and can capture that long range interactions as well. So we have model all those different scenarios, taking non-context into account, taking local and then long range context into account, and have compared models. So maybe in future talk, I can talk about that part of work. So we have done extensive experiments in that area, and have some suggestions on what could be the optimum combination of tools to use. Yeah.
Okay, thank you.
If there's no more questions, I'd like to thank Sonia again for your time to give this wonderful talk. With that, thanks for your time attending this TechTake talk. We probably can call it a day. Thanks, Sonika.
Thank you so much for having me.